Hello and welcome yet again to another very entertaining video. This video is meant to be a continuation of the previous video regarding female reproduction, in which we discussed folliculogenesis. And we mentioned that it's often confusing to understand the difference between oogenesis and folliculogenesis. To kind of highlight those differences, I'd like to start in the same place, and that's going back to the primordial germ cell and how during embryonic development of essentially the mother, this primordial germ cell is wrapped up, surrounded by somatic support cells, and essentially this PGC goes into hiding, right? This cell right here represents the next generation or the children of the developing embryo. In this stage of dormancy, it's called a primary oocyte. And it's called a primary oocyte because it's actually entered into meiosis, and it becomes arrested in prophase of meiosis 1. And this is where we want to pick up the story, because it will not awaken from this arrest until after it's been ovulated. So, to understand ovulation and the ovarian cycles, now we have to talk about hormones. Our previous video detailed the hypothalamic pituitary axis. We showed GnRH regulating FSH and LH and estrogen and inhibin, and how all of these things were regulated. We now want to provide some timing. So let's build our timeline, and let's look at this from the vantage point of a typical 28-day ovarian cycle. We'll start at day 0, we end at day 28, and there's day 14, which is about halfway. Now, what I want to do, I'm going to draw 4 different colored lines. And as I draw them, I want you to think about which hormone is represented by that line. There's line 1, there's line 2, there's line 3, and there's line 4. So what hormones should we assign to those lines? In green, we have LH. In yellow, we have FSH. In blue, we have estrogen. And in pink, we have progesterone. Now, as we continue where we left off from the last video, through folliculogenesis, we created, over a period of 5 months, a mature Graafian follicle. Now, this follicle was characterized by a primary oocyte. And remember, that primary oocyte has been arrested in that same phase. It hasn't changed for years, since this female was still an embryo a long time ago. And it is surrounded by granulosa cells. We actually have the immediate surrounding there that we call the cumulus oophorus. And then more cells out here, we call this space the antrum. Remember, we're still within the ovary at this point. Now, to kickstart the further development of that primary oocyte, to have it continue through meiosis 1, the signal for that is the LH surge. And as green is our LH, we see this event right here. Now, I want to take a step back and ask this question. What causes the LH surge? We talked a little bit in the last video about how estrogen has this biphasic effect on some of these hormones. And while this is an oversimplification of the process, I think it's easiest to think about it in this way. The blue line is our estrogen line, and at low levels, estrogen is inhibitory to LH. In other words, at low levels, LH is blocked. But remember, estrogen's gone into this positive feedback loop where estrogen is driving the production of more estrogen. And I want you to notice that the highest point for estrogen in this cycle is right before the LH surge. Here's the estrogen peak right here. And the reason that's important is because high levels of estrogen, rather than inhibit LH, now stimulate it. And so we'll see the highest estrogen peak right before the LH surge. And the LH surge is caused by the massive amount of estrogen. Now, the LH surge is going to do 2 things. Number 1, this is the signal for ovulation. I know that within 34 to 36 hours after the LH surge, we're going to get ovulation. And the mechanism for this, if you recall, one of the things that FSH did to the granulosa cells was cause them to begin to upregulate LH receptors. And following the LH surge, the granulosa cells are now responsive to LH, whereas they weren't before. And that's what induces or finalizes the ovulatory process. Now, the second thing it does is it awakens that primary oocyte. The LH surge causes the primary oocyte to continue meiosis 1. And with that comes a cell division. So now within the cumulus ovarus, we would expect to see 2 cells. Now notice, this wasn't your typical cell division. I don't have 2 identical daughter cells. Through mechanisms that aren't completely understood, one cell is chosen to be the dominant oocyte. The other cell essentially gives everything of value to that dominant oocyte, essentially sacrificing its life and becoming what we call a polar body. So this is our first polar body. Under normal conditions, this cell is typically going to die off fairly quickly, and we don't have to think about it too much. But now the oocyte is called a secondary oocyte. However, it doesn't complete meiosis yet. It arrests in prophase of meiosis 2. Now we undergo ovulation. The Graafian follicle is going to rupture, and the cumulus ovarus containing these 2 cells is going to merge onto the surface of the ovary. Now, if we follow that, the fimbriae of the oviduct, through peristaltic wave-like movements, will literally scrape that oocyte surrounded by the cumulus ovarus off the surface of the embryo and pull it into the antrum of the fallopian tube, where fertilization is likely to take place. Now, it's important to recognize what happens to the remnants of the follicle. The follicle becomes the corpus luteum, and whereas prior to ovulation, the key hormone being secreted by this follicle was estrogen, we now start to see a transition. The cells convert from producing estrogen now to producing primarily progesterone. So let's zoom out and see what that looks like on our graph. We can see here at day 14, estrogen levels have been decreasing, and then we see the rapid uptick in progesterone. Progesterone then plateaus off and then starts to fall off just a little bit later, after about a week and a half. Progesterone is important for really 2 reasons. It has 2 jobs. First of all, progesterone's main job is to maintain the uterine lining. The importance of this can't be understated. RU486, the day after pill, this was a drug initially designed to treat Cushing's disease because it blocks receptors associated with cortisol. But similarly, it blocks progesterone receptors. And if I lose the progesterone signal, even if implantation has taken place, the uterine lining will slough. The second thing progesterone does, and we talked a little bit about this last time, it strongly inhibits GnRH. As such, as long as there's progesterone in the body, GnRH is inhibited and there will be no ovulation because there won't be any FSH, there won't be any LH, and the ovulatory cycle stops. And so now we start to come full circle. After about a week and a half, progesterone levels will start to decline. We see that here. With the decreased levels of progesterone, the inhibition on GnRH starts to relax. And that leads, at about day 26, to this increase in FSH. And that's really where we start the cycle. As FSH increases, FSH recruits a new Graafian follicle, and we go back to day 0 and start the process all over again. Now, what about pregnancy? What if an implantation occurs? If there's a successful implantation, a round-a-ball of cells called a blastocyst will have already differentiated into an inner cell mass and then an outer cell mass called a trophoblast, which will become placenta. The trophoblast starts to secrete HCG, human chorionic gonadotropin. And that HCG really just has one purpose: to signal onto the corpus luteum to tell it, hey, we need progesterone. This maintains progesterone. As such, we would see progesterone continue beyond day 28 rather than falling off. This maintains the GnRH blockade, and ovulation will remain on pause for the next 9 months throughout pregnancy. However, if that HCG signal is never seen, then the corpus luteum involutes essentially dies off, and it becomes the corpus albicans, which is essentially a little pockmark, a little scar on the surface of the ovary, and is no longer biologically active in making hormones. And you'll probably recognize HCG. This is the hormone that pregnancy tests are looking for. So it's found in the urine 7 or 8 days after fertilization and can be easily measured with a quick pregnancy test. All right, to finish up, I'd like to go back to progesterone and its role in maintaining the uterine lining. So we can juxtapose onto this 28-day cycle the uterine cycle. And keeping in mind the hormones from the ovarian cycle, we're going to see those hormones playing a key role in what's going on within the uterus itself. Keeping with the same calendar, we've got day 0, got day 28, and right here in the middle, roughly, day 14. And we can break this cycle down into 3 different phases. From day 0, roughly to about day 6, we've got menses. From 6 to about 14, what we call the proliferative phase. And 14 to 28, the secretory phase. Now, going back to day 0, how does this process begin? Well, remember, it began with the loss of progesterone, as progesterone maintains the uterine lining. In the absence of progesterone, the thickened endometrial lining dies and sloughs off. Now, this is important because of the amount of energy required to maintain this lining. It's not something that the body wants to continuously do. We also want to keep the lining new and in a state receptive to the possible fertilization or implantation of a fertilized egg. So, through the first 6 days, the loss of progesterone causes the death of the endometrial layer, and it sloughs off. Now, the proliferative phase, this is when we start to rebuild. And if you think about hormones, remember the dominant hormone during this phase is estrogen. And it's estrogen that's driving the growth of new endometrial tissue. Then, following the LH surge, we have ovulation, the corpus luteum converts over to making progesterone, and with that we enter the secretory phase. And as we prepare for possible implantation, there are really 3 things that I want you to understand about the secretory phase and what progesterone is doing. We see a thickening of the endometrium, we see dilation of the glandular epithelium, hence secretory phase, and lastly, enlargement of the blood vasculature, all of which are again required to help prepare the uterus for possible implantation.